We got everyone unless somebody else joined. But yeah, thank you all for coming. We hold these every other month, just to kind of review jumping worm projects and updates and regulations, all sorts of stuff. But today we're going to talk about a handful of things, including this upcoming symposium that our lab is planning on jumping worms. And Vera will take the wheel from here. Vera, are you good to go?
Yeah, I'm good to go.
Awesome.
I'm glad to see so many people had the time to commit today to talk together. I know this is a very busy time of the year. Every time of the year is very busy, so we appreciate your attendance. So what we'd like to do is go around to start with and have people give summaries of, sound bite of what's new. And, I would like to end it because I would like to get consensus about this symposium we've been planning with Joseph and Shannon and Brad. We talked to Brad yesterday. So we can do that at the end, but first, can everybody from every lab
Looks
Maybe
like,
I can.
oh,
Oh, no.
Vera's cutting
invite
out.
you, Erin?
Oh, are you back? Okay, cut out a little bit.
Erin, how about you? You want to start?
Sure, I can start. Yes. So, I actually just met with Kyungsoo today, along with his graduate research assistant Nate. We have been working on a manuscript for Frontiers in Forests and Global Change on developing a soil plant continuum, a method for detecting and quantifying damage from jumping worms specifically, but could be used for other soil dwelling pests. And so, yeah, it's a perspective article. It's small, 3,000 words, but basically kind of using the current sort of data captures of like, Sentinel Plant Network or public gardens as sentinels for invasive plants, how they are using data to help define whether or not something is invasive and kind of really specifying the impacts on soil and how those impacts on soil also impact our plant communities. And so we just got our review back with really helpful, constructive feedback. And so we're going to submit our comments back to the reviewer and then hopefully make some revisions and then get that finalized for hopeful publication. And then another thing that I've been working on just recently was I was invited by the American Public Gardens Association to work or to submit a poster for their poster session, talking about the different jumping worm research that is happening here at the Minnesota Landscape Arboretum. And we thought that that would be a good opportunity to revisit the survey that Joseph, Brad, Brandon Miller, Miriam, and I, and now Kyungsoo are going to be a part of. We mostly want to survey these public gardens to, one, find out how aware they are of jumping worms, find out if they have them, where in their gardens they're noticing them, have they disappeared, how did they learn about them, what are their visitors asking, if they're asking questions about them, those kind of questions. So using this poster session at the APGA conference to catch attendees and get them to the survey up on their smartphones and actually submit it right then and there if we can. And that way we can because the last time we tried submitting it through the APGA newsletters, we got like 15 responses. We can do better, especially if I can corner them and force them to take the survey in person right there with me. So
Yeah,
that's kind of
forcing
where we're
them
at.
works.
Yeah, yeah. So that's where we're at right now.
Anybody have any questions?
That's you froze up, but yeah, if you have any questions for me on either the perspective or the survey, let me know.
Frozen before. Can you hear me, guys?
Now we can.
Yep.
All right. Okay, so thanks. That's a lot going on there between you and your collaborators. So thanks, Erin. Joseph, do you want to go next?
Sure. Um, I don't really have much to report because it's winter, right? So, not that anybody else wasn't in the same position in Minnesota, but um, yeah. So I'm going to start my 16th year of surveying a population of jumping worms at the University of Vermont's Horticulture Research Center this actually next week. Find out whether there's any worms that have hatched, and that'll go on until November, I guess, on a biweekly basis. That's what I'm preparing for right now, and I'm just coming out of um, uh, being a chair of a search for a soil ecologist, and we certainly have taken note of people's experience in working with jumping worms in that search. So hopefully we're going to have another collaborator pretty soon. And I guess, maybe I should say something about Vera's push for another symposium. I think that is a really great idea. I think especially when at this time of year. So if we can get this done by June before everybody panics when they see earthworms out, that'll be really good. I guess from that point of view, I should also mention that, and probably some of you have probably been approached by I think it was Washington Public Radio. They were worried about, or they're going to have a feature in May about jumping worms in Oregon and in Washington. And so if you haven't been approached yet, maybe that's something where we can help them out a little bit more. And if we have a date for a symposium, maybe they're going to maybe we can have them fold that into their just announcing that symposium to their listenership.
Yeah, Joseph, I invited them to our last blog. So they
Uh-huh.
were with us March 2nd.
Oh,
And
cool. Great.
and then they took some of the people's names, and I don't know if they said they would further contact some people. So I
And
have
they
their
have. Yeah.
name, so I definitely will do what you just suggested and tell them what the group is working on for public education. That's a great idea.
Yeah, for public education, and then also this symposium might be a really good place to advertise the symposium as well.
Perfect, good
Hey,
idea.
hey, Yosef, this is Angie Gupta, and I have been thinking about you.
Oh
Really,
no.
I recall that you were a part of an effort or aware of an effort, I think in Vermont, to get one of the pesticides relabeled for worms. And I'm curious if there's an update to that.
Yes, there is. So update 2 years ago, I was on sabbatical in Canada, and I confidently said that Bovaria bassiana had been approved for use on earthworms, and by the Department of Ag, by the right people. And then I get a nasty call from the head of them, and he said, "Well, that is not true. No way would we have allowed you to do this, or I would have allowed my people to do that." And I said, "Well, they did, And so and then he said, "Well, you can forget that. That's not going to happen." So there you go. That's my update, right? So, and I said, "Hey, Section 2EE, what's your problem?" So, well, we have Vermont, we have our own rules, and they do, So I mean, they have very stringent. We have very stringent pollinator protection when it comes to pesticides, and for good reason, right? I mean, I'm not saying that there's no good reason, but I've taken this really strange position right now since I've been shut down, and I was not the only one who got shut down in the state, but on a similar charge. You don't have any right to even do any of that, right? So, but I've come to this position that it is really difficult, right? So if there's a new invader, that's a bit like a new designer drug. Those people that the regulators are always one or several steps behind the invasion or behind the designer drug, right? So it's not on the list, then there's nothing they can do until somebody puts it on the list, and it's really their decision to do that. But Vera said something really cool at our meeting in June. Said, hey, you guys at the Healthy Soil Collaborative keep doing what you're doing, because anything that you can find out about these worms and about the damage that they do can be a nail in their coffin, right? In terms of convincing regulators that it is necessary to do this. The reason why I was given sort of a falsely given the go-ahead for Beauveria bassiana 2 years ago was because the politicians in Vermont and the regulators in Vermont got a ton of phone calls from the public saying, you've got to do something about these worms, right? And so they said, you've got to do something about these worms. And so I said, well, how about there is this microtized millet that you can use, and it works pretty well. So why don't we have that approved? Because it's already approved for other pests, so that's that 2EE thing, and the millet shouldn't be regarded as a new pesticide, right? So it's part of the formulation, and so it should not be a problem. And so I said, yeah, that sounds great. Let's do that. And so they did that. And then, as I said, I got the nasty call from the leader of the pack saying you can't do that. But I'm not the only one. They're pretty hot on the trail of offenders. So we have a guy here in Vermont who does gardening podcasts, Shalina Dosi, and he at some point said, "Oh, the way you can protect your plums from this particular insect pest, you can spray the plums really early in their development with kaolinite clay." And so he had the same thing. He got a nasty call from somewhere saying, well, what you are saying is that you will put that this clay is a pesticide, and you have no right to say that because you're not even a certified pesticide applicator. So they're pretty hot on everybody who wants to do something about anything, really. It's difficult. I think that there are some states that are much easier to deal with. I looked at the Washington and the Oregon regulations for the minimal risk pesticides, and it's a very simple approval. It's just you pay some money and you fill in a form, and it seems to be okay. So I think it'll be really cool, and maybe Vera has done this already, to have a look at the mosaic of regulations across the nation. What do states really require, and how much effort is it, and who makes a decision in the end.
Thank you for that update. I'm sorry.
Yeah, we can talk about that later. But Joseph, kaolin clay is now approved as a foliar under 25B. So you got to get back to
Sorry, which one is it?
Kaolin clay is now approved. I just looked it up as a spray on fruits.
Yeah.
Organic
but
is
not in
25B.
Vermont.
Okay, not in Vermont. All right. So that is true. Every state has the right to be more restrictive than the federal regulation. So that is absolutely true.
But it'd be kind of cool if every one of us takes 2 states or something like that and inquires what it takes to have minimal risk pesticides or ingredients approved as pesticides, or the 2EE regulations applied to a pesticide. That'd be really cool, right? Because we could then make a big table and saying this is what needs to happen in these states.
So kaolin clay is a 25B, so it doesn't need a
25B. Okay.
So 2EE is for a pesticide that had to go through the regular registration process, and then that costs like $1 million. So these novel organic pesticides that are somehow based in nature, so kaolin clay is used in food. It's a additive to many foods. That is an organic, and I don't know if Jim will talk about it, but we've been basically going back and forth with this over an issue. And there's two tables, and the first one, table one, table two, and you have to have your active ingredients and your adjuvants on those two tables to get a 25B registration. So a
Right.
25B registration like costs $500. And
Right.
so it would be a great way. So the EPA gives those out, but then the state has to approve it.
Right. Yeah. I think that's pretty much it. It'd be good to know. So that was similar to what they were doing in Washington and Oregon. The around $500 and a form for the 25B lists.
Anybody else with any questions for Joseph? So Joseph, I agree. I work on Japanese beetle too, and they're both soil inhabiting like jumping worm. And even for Japanese beetle, which is such a huge environmental and economic pest, there's very little dollar value on what the damage causes. And that's what you need to convince anybody to put it on an important invasive species list that you're going to get money from. So the good news is the new federal grants, the new NIFA grants, have come out, and for us, invasive species are getting the most money for grants this time around. So a
Wow,
lot of
cool.
it's invasive species and harmful additives to food or national security issues. So those three issues: food additives, invasive species, and
National security.
national security are the ones that NIFA has now said in the next few years they're going to get the most money. So anything we can do, just keep everybody's radar to quantify environmental and economic risk is really important, because you put that in the front of your grant and reviewers who are not convinced jumping worms are an issue say it is. So one of the things we did is in the handout today, there's a picture map of the United States, and we've put every state that has different policies about jumping worms. So you can use that. You can say of the United States, of the states, this many have it restricted, this many have it on whatever different status. So that will help you a little bit, the status of its invasiveness by state. But it would be nice to have some economic dollars to show what is going on. And I guess that's the research you guys are doing right now, trying to figure out, like for the maple industry, how this is affecting seedling establishment.
Yeah. So I mean, I have talked. I've offered to do some of the economics. I'm not an economist, but I've worked with natural resource economists, and I kind of know which way they go. And if I come up with some kind of objective function and I throw it at a natural resource economist, they might actually become excited about this kind of thing, right? Because we know that the amount of money that, or the dollar cost of invasive species invasions is about $2 trillion over the last 20 years. So that's a lot of money of damage that's been caused. And of course, not all jumping worms. I mean, I wish we could say that, but it'd be kind of nice to have the dollar value on a jumping worm. So here's the jumping worm. This is how much it cost you. Right? This one worm cost you 50 cents. And then you can extrapolate up, right? But there are just so many different aspects to it. Like, so there's the cost, the direct damage. Things like water quality and what's the cost of providing bottled water, for example, compared to taking that water out of a contaminated water supply. There's the amount of water, right? The amount of water that you can get. Like, let's say you have a reservoir, then that reservoir is being impacted by sedimentation, and so that takes away from the volume of that reservoir, and that takes away how much can be taken from it sustainably. That's water per year that can be delivered. So that's another cost. But the maple industry probably a longer term issue, but it is there. I think the big question, or the big problem in doing this kind of research is to be able to integrate over space and over time, right? So if you take space, and you have all these different kinds of economic concerns that are within a region, but the impact might not be felt today, but in 20, 30 years time, right? So we have to kind of forecast the cost of the damage that will be showing up at a later date. And then there's a whole bunch of costs that are really difficult to quantify, right? So things that have to do with heritage and culture, like what about how much more fun is it to hunt a deer when there's no understory, right? So, and how much? I mean, I'm not a deer hunter, don't worry, but how much less fun is it to do that, right? So a lot of the hunters will tell you that they really enjoy stalking their prey for months ahead of time, and it would probably not be as much fun if they had to just go through a forest without hobblebush, So that's just one example of that, right? So there may be other things like mushrooming, right? So what are the mushrooms doing in those invaded forests? Maybe some of you have looked at that. What happens to mycorrhizae, and what kind of effect does that have on particular plants that are used by indigenous people? So there's all this cultural thing that is really difficult to valorize, but would definitely have some kind of effect on people's psyche, and enjoyment of the great outdoors, right?
So I have spoken to two. We have in Minnesota, we have Native First Nation reservations. And so I've spoken to two different groups about jumping worms, just like you're talking about cultural heritage and also its impacts on understory. And they're very concerned. And so they actually came to our last meeting, one person, and then I followed up talking to her personally. So there is a concern about jumping worms on these native lands and the effects on understory so far. And then the next person I talked to from a different group, he was seeing effects in his, as you said, in his heirloom garden on plants
Mm-hmm.
that they were growing. So yeah, it's the thing is we have to get a paper with this in it, and then people can use that as a reference. Right now, it's
Yeah,
just hearsay. Yeah. And maybe
yeah. I
that's
think.
what I will work on for this symposium. I think that is we've done a meta-analysis in our lab. We sent it to you. It's rather long, and we can now ask questions that meta-analysis, ask questions like this: how many paper shows there's effect on recruitment, effects on what CNN ratios, and extrapolate that, and we'll call it environmental and economic impacts of jumping worms. And then now we have a reference. So I will take that on, Joseph, as
Oh,
part of the
cool.
symposium. Yeah.
Yeah, I think that is, to funders, that is a more it's a less tangible thing in terms of economics, but for funders that might actually be very attractive, because you're dealing with heritage and with culture and um, and emotions, right? So that goes with emotion. I'm from Germany. We love our forests, even though they're horribly clean because people pick up wood and stuff that fell down. But the Sunday afternoon stroll to the woods is culturally really important. Strangely, don't ask why. I have no idea. But so if the woods disappear, then that would be a big deal.
Yeah. Thank you, Joseph, for bringing those up. Those are good things to think about. Who else would like to go next? Angela, do you have something to talk about?
I think I will generally yield my time to others. We're ramping up our participatory science. Hannah will maybe talk about that and doing a lot of social media, but I'll yield my time. Thanks.
So, Angela, I wanted to quantify this and then I lost track. You were recently in a University of Minnesota release about your citizen science program on jumping worms. I read this somewhere. Can you tell me where I read it so I can put it in my references? It gave accolades to you and was talking about the success of getting citizens involved, and it used your program as an You don't remember this, huh?
I don't think I know this. I would love to hear
It
it too.
just happened since January, and it was something CFAN's released. So I'll have
Okay.
to go back through their newsletters.
I'll see if I can, I'll look through my files and see if it's not sparking, and then I'll drop it in if I find anything.
Thank you so much. I appreciate
Yeah.
that. So,
Yeah.
some of us met back in September to talk about how it was at Joseph's request, and we were trying to figure out the distribution in the US of different amanthus and related species. And so we had a meeting with somebody Joseph knew from New York State, right, Joseph? Maybe correct me if I'm going to say anything wrong here. And we got to talk about EDDMS maps and iNaturalist and how to get people to post stuff. And then in the process, we realized that we don't have really good keys or really good for morphological identification or really good ways for chemical DNA identification. So people could say they found a wiggly worm, and then they would have to send a sample to somebody to identify it. So we could make a distribution of jumping worms as a group. That's fine, that's important too, but that we really need better diagnostic tools that we can give to the public. Does everybody feel that still is an issue? If
Um,
Yeah,
and
go.
I think Anis Dobson was saying that that iNaturalist now has some kind of AI thing that can identify um, jumping worms, not necessarily to species, but just the whole jumping worm complex. That might be helpful. I will send Anis a message with Vera CC'd in so that the response goes to both of us on that.
And I have a paper that was, I have an IPM manual online, and there is somebody in our college in the Department of Bioengineering, and her name is Si Yang. And so I helped her grad her PhD use AI on my IPM manual, that's like 350 pages, to see if they could use AI in photographs to match it to one of the 300 identified pest pages I had. Okay. And so after this meeting in whenever, I think it was August, um, with Anna Dobson, I went back to Si Yang and I said, look, we have this issue. Could you help develop an AI key for jumping worms? And she said she would love to, but she needs pictures and she needs a morphological key, and she has some NSF grants on this. Taking ways of done again, Department of Defense, everything. You go to Iraq and a spider bites you, and they want to put the spider on a scanner, take a picture, and tell you if it's toxic or not. So there's a lot of money out there for this. So she has money, and she would love to help us. So maybe we could try to figure out what subgroup of people to talk to her about this.
So this is Angie. I dropped in a link to the chat, which goes to the iNaturalist um, article, for lack of a better word, which I think Laura discovered this actually. But this is a really great ID tool, and I what I think I heard Anna say was just how good iNaturalist has gotten at its artificial intelligence using photo recognition of worms. I feel like I heard her say it was 100% accurate in her work in New York City. Yeah, so I mean, I would iNaturalist is a super awesome tool and it does tons and tons of things. And if it can do the thing we want, I don't think we should make another tool. It's
Okay,
kind of where I'm
so
going.
yeah, that's a good idea. So maybe since Anne is the expert, I will talk to her and see. Yeah, I'm on the page that you gave, and it does have some. So
And for
that's
what it's worth, I mean, I'm an iNat ambassador and a very active iNat user. And iNaturalist has been good for a long time, but it is very good at photo recognition at this point. I would say it's gotten very, very good. And so you have to have good quality images to do worm ID, to do many types of ID, but worm ID included. So, but that's a user error issue, right? So, I mean, bad pictures are not identifiable by us or by AI. So, with good pictures, I actually have a pretty high degree of confidence that iNat can do it for us.
so has this changed the distribution? Has the output changed? Is the distribution map better now of jumping worms in the US?
I feel like Laura's on this call, right? She monitors EdMap's. Wondering if she's gonna, yeah, jump in here.
Yeah, we import iNaturalist reports on jumping worms from Minnesota into EdMaps, but I've not done a lot of looking at jumping worm reports from other states in iNaturalist. Uh, so don't really feel like I can speak to how accurate the INAT reports are from Ohio or something like that. I think you'd want someone with more species ID.
So say
And when
that
you say
again,
it's so
Laura.
good, like, are you saying that like it can tell Tokyoensis from agrestis by a photo, even if it's just a worm on dirt and doesn't have close up of the pores like that?
No,
That
I mean, so you
sounds
have to have the
suspicious.
right. I mean, I mean, it might give you that answer, right, inappropriately, and then in theory, it won't get through the second level of human verification to become research grade and thus output, then pulled out. I mean, we know INAT is not perfect, right? And if you don't have a picture that shows the characteristics, then it can't, like, none of us are getting it right. With a picture that shows the right characteristics, and it's multiple images that you're putting into iNaturalist. I think it's most important to have your most identifiable image as your first one. I think it's running A on one image, not on all of them. And then the human, I know when I look at them, I commonly look at many of the images, right? To compare and contrast and look at angles and all that. So anyway, but my point was if INAT can do it, I wouldn't create another tool. And I
So
suspect INAT
Can
can
someone
do it.
test it? Like if you have
Yeah.
photos of five different species, can you put them in? Like if they're just a regular photo of a worm on a dirt, like is it really giving you accuracy by that species level, or is it at the, family level?
It's a great question. I mean, the answer is you could totally test it. I don't have access to all those worms
so
to do
is
it.
somebody testing that? That's my question. So to me, iNaturalist, I really don't know how to get data evaluation out of it. So people can use it, as you said, to identify, maybe to some degree. We have to talk to somebody who actually uses it. But can you, as somebody who isn't that individual, go there and get information on jumping worms from there?
Yes.
Okay, so I'm gonna talk to you privately on how you
Yep.
do that. So you can get a list of all the people who put in worm images and the output they got.
Yes. The searching, yes. To get that type of data, you're probably exporting it as an Excel file, and then you can see the reporters. There's different ways in which you can search, but the short answer to those questions are all yes.
And then somebody verified that the identification was correct.
Only if it's to research grade. And I'm looking right now at the worm ones, and at least, maybe 50% are to research grade as I scroll through the first page, which is, I don't
Yeah,
know.
so Vera, you can see both reports where there haven't been enough verifications. It might just be at the Amenthus level. And then you can also see the reports that have maybe been made to the Amenthus Tokyoensis level or what other levels you're looking at.
Okay. Well, I'm probably going to contact one of the other of you, and if you can walk me through to how to get this information, I would appreciate it.
Yep.
All
So I
right.
And I
just
know you
wanted
encourage
to,
volunteers, if any of you more research people who have beautiful pictures of different species of Amenthus would be willing to just try, if you have an INAT account, if you'd be willing to try uploading your pictures and seeing what the INAT AI suggests and just like informally reporting back to us, I think that'd be really interesting.
That
And I
seems
see
like a great Europe proposal for somebody.
Well, and for what it's worth, you don't actually have to submit the request to see what it's going to give you as choices. So you could do this without screwing up the data too.
so Vera, I just wanted to get back to your other point about identification. So I'll introduce myself because this is my first meeting here, but I'm Carrie Dean. So I was a postdoc in Vera's lab. I'm now an assistant professor at University of Alabama, Huntsville. And my grad student, Katie, is also on the chat here. So she's a master's student who's going to be doing a project on jumping worms down here because, as far as we can tell, no one's done any work in Alabama on them, despite our state being like 70% forested. And so she's going to be working with molecular techniques. So we're going to be trying to develop a soil eDNA technique for being able to test soil to see if detect any potential jumping worm DNA. But it's all based on that Nori Ain paper where they develop primers for the three species. What were they again, Katie? So I think it's Tokiensis, Agrestis, and then Metafire,
Yes, it was those
I
three.
believe. And so she's going to be kind of playing around with that. So that's basically it uses, PCR to detect species, but we're also thinking about in mixtures, maybe doing something with qPCR. I'm not sure how important that would be. But we're just saying if any of you have samples that you would want us to, kind of test, we'd be willing to do that, certainly within the group. So Katie can get really good at it. Um, and then also, I mean, we're going to be doing some sampling down here, at least in Huntsville, so in urban potential source nursery sites and botanical gardens and things. So I just wanted to throw that out there.
Well, we'll send you. We now have a second jumping worm species that's coming with the worms, the composting worms we order from Georgia, and it
Hmm.
behaves differently. It does not act the same, and it reproduces much faster.
Mhm.
Grima, who's going to talk, and we have no idea what it is yet. It is a jumping worm, but it's not the same species because behaviorally we can tell that. But they're juvenile, so. We don't know how to identify it yet. So we'll send you some samples.
Okay.
I was going to say, Kiangsu had his hand up for a
Uh,
while.
yeah. Oh, thank you, Joseph. I do think this identification is a real problem, even after iNaturalist does a good job with the photos. What we really need is a really refined three-dimensional model that allow us to reproduce in the plastic forms with the 3D printings. We thought about this with Hannah and Angie. Um, but if any of you know a medical person in the University of Minnesota Hospital or a Mayo Clinic who do micro CT, then we can collaborate with them and basically get the three-dimensional model of different earthworms to a really fine scale that we can actually touch the setae and then all the pores and the segments. And that will really revolutionize teaching, the identification, and the morphology. And even for us, and it will be really helpful because even though I'm a jumping arm researcher, I don't really have a skill to tear apart Tokyoensis, Agrestis, and Metaphyr Hilgendorf. So it's a shame, but then it's a real problem, and I don't really have a skill to go between the real specimen in my hand with the Chang et al. Papers to really compare those things and then get the species right. So yeah, and so I think one thing we can probably collaboratively do is to think about this a three-dimensional model.
Kingsley, I've heard you say this
Mm.
request before. And what is that technology or what is that medical? I don't know what that
Oh,
is.
that's a micro CT. So I will put two links on the chat. So the first link is about all the animals that Natural History Museum, for example, is going through. And the second one is actually a paper about microcity of earthworms. So my computer is going slow. So I will drop the second link once it come out.
Because I have
Mm.
made 3D print models of other invasive plants mostly,
Right,
and
yeah.
my colleague Megan
Mhm.
Weber in aquatic invasive species has quite a number of like fish on a stick and other things. And the artist has changed her plastic so they're a little bit more pliable. And she gets like fish models, for instance, down to
Mhm.
scale level accuracy. So there's the right number of scales on the side, and then she hand paints them. So um, I have been involved in working with her a couple of different times, and it can be a lot of work to make sure that she's representing it correctly. So there's a lot of
Okay.
back and forth between like people that
Mhm.
know worm identification and Carlin.
Uh-huh.
But I think that we could do without needing access to this medical infrastructures is kind of what I'm wondering.
Mm.
And it's gotten pretty affordable. You pay for like the
Mm.
amount of plastics you use, and worms aren't very big. So, I mean, I think if there
Yeah.
was interest in doing this, we could get it done.
Yeah. Guess, probably I need to understand how your collaborators, produce the 3D model. But what I'm thinking of is basically the coordinates, three-dimensional coordinates of the surface of the earthworms in real fine scale that can be downloaded from computer
Hmm.
so that everyone can reproduce in a size they want.
So the
So,
way, and
yeah, yeah,
I
yeah.
don't know how long Vera wants to talk about this, but the way she does it is she develops it as a sculpture in a
Sculpture, yeah.
digital medium, right? So as an
Mhm.
artist sculpture online. And so she can get down to like segments and see that. Now, I have to
Mhm.
say the hairs worry me for like, could they break off, right?
Mhm.
That's a challenge that we've had with like petioles on long branches that can't support the weight of the leaf, right? So there's some things there. But I mean, she's been able to make it work with like fish fins, right, that have to attach to a small
Mhm.
place.
Mhm.
And so she does that in a digital model median. And then as experts, we review that. And then we're the ones that are like, okay, do you have the right number of segments or
Mhm.
CD or whatever? And she then makes adjustments. So it's a back and forth, back and forth.
Right, yeah.
And I don't know if it's better or worse, but that's the process that I know.
Mhm. Yeah, looks like it has a somewhat different process from collaborating with CT scanner, an operator of the CT scanner, because then the operator of the CT scanner can actually scan the live earthworm specimen and create the model. And then, but in your case, then your artist basically render the three-dimensional
Mm-hmm.
image in consultation with you. So those two are different.
Yep.
But I think there could be really good collaborations across all levels. So if when we need some data that some morphologic data that can be reproducible, then this CT scan based ones could be useful. But then between this scientific accuracy versus public's acceptance of those models, I think there is also a genuine place for artists who can create the three-dimensional models with clay or other mediums, and also encourage the publics to create their own. And for example, when it comes to identifying, nightcrawlers versus, amensas, we don't really need to go into very much details, right? So some simple features will justify the differences. But let's say we need to go down to, differentiating agrestis versus tokyoensis, or, amensas versus metaphyre, then those tiny bits become important. And then that's when probably this CT scanner can play the bigger role. Yeah.
Yeah, so if we
And
can
yeah,
come up with
yeah,
somebody with a
yeah,
scanner, that's
yeah,
great, right?
yeah.
Like,
If we can find someone, the reason I think that is feasible is because there are some studies in soil science in which CT scanners are used to map out the soil structure. And then in those papers, often there is a collaboration between soil scientists and hospitals
hmm.
who has CT scanners. So I think it is feasible to form a collaboration with the UMN Hospital or Mayo Clinic. And since Angie, you're in Rochester, then then we can. Yeah.
So I'll just share my
Yeah.
limited experience trying to do like 3D model work with
Yeah.
hospitals is I'll just tell you their rates are extraordinary. So like to get,
Oh,
because they're looking at like reproducing hearts or things like that.
yeah.
And so they can just like spend lots of money. And I'm like, I I got a plant. And so
Yeah.
it might work. I don't know anyone.
Yeah.
I'll be like, to get resources at the Mayo Clinic or to collaborate is like a whole challenge.
Okay.
I'd like to think the university has their own hospital, that we'd have access to that, and the medical school, but my experience is not quite in line like that.
Yeah. Okay. Because I just saw that Mayo Clinic made a huge surplus last year, so they might become more generous. But
Yeah, no, that was less than a previous surplus, and their generosity
okay.
has not increased.
Anybody else want to continue this discussion? I think it's fascinating, and I hope somebody works on it and comes up with some good identification tools. Anybody else? Katie, do you want to talk about your master's?
Yeah, I could do that. Just like a mini run through of the proposal, maybe?
Good idea.
Okay. So I have three objectives, and my first objective is to go around to urban areas. So I will look at wholesale nurseries. I will be looking at the botanical gardens and their ornamental beds, specifically ones with Asian native species. I will be getting samples from bait shops, or I will just purchase, bait for fishing and then run DNA extractions and then PCR to determine if there are jumping worms in the bait. And this is just to identify possible, um, I am forgetting words, methods of introduction. There we go. Sorry. My second objective is to set up a mesocosm experiment where we will have different containers with some with, or one with no worms, some with each species of jumping worm, and some with jumping worms and European earthworms together. And then I will do two eDNA samples on those, one while the worms are alive and active, and then the other one after the worms have laid their cocoons and died. And so with that, we are hoping to kind of narrow down a soil eDNA technique. Then we will survey state parks within the state and use those soil eDNA techniques to see if we can detect jumping worms in the state parks. And I will specifically be looking at areas where they have landscaped and introduced non-native species and where people like to go fishing in those state parks. So that's a quick rundown of my proposal.
Well, that sounds like a lot of work for a master's, and I think what you're doing is really important. So hope you can sort out some DNA
Thank you.
answers. So remember that all these really good professors on this committee, on this, call it a community of practice. They might be able to be on your committee if distance delivery or something. So don't rule that out if you need to get some more expert experiences and expert information from other people. If
Thank you. I really appreciate that.
if there's, it seems what you and Carrie are saying is you don't have a lot of colleagues down there. You're not in an agricultural school. You're in a basically medical rocket science school.
Yeah,
So, yeah.
engineering is the bread and butter here.
So you don't have a lot of colleagues who know about biology. So I don't know what people would say, but there's a good smattering here of researchers who might be able to help you.
Uh, can I just, I think that you guys already know about this undergraduate student in Maine that did a thesis on eDNA and looking at stuff and looking at distribution of worms in Maine, and she was not very successful, but successful in making it work. But she had some suggestions, so just shut me up if you already know. I think she's on your list of people for the symposium.
Yeah, we have her paper in our meta-analysis. Yeah,
Cool.
yeah.
Great.
Yeah, thanks for that. A big part because Katie's main objective is really focused on developing an eDNA technique. We're testing a couple parameters. So testing, in terms of extracting DNA from soil, different amounts of soil. Also, this mesocosm experiment is meant to kind of create these artificial combinations and see how effective, sampling some soil from those. So we're trying to kind of optimize that a little bit in ways that maybe would have been helpful for her project. What else are we doing? We're also looking at, oh, there's something else I can't remember. Just to kind of try to optimize that and then kind of
Mhm.
test it to see if we can detect worm DNA in sites of known infestation. But yeah, in terms of being able to detect jumping worm DNA from state park sites, I don't know how realistically successful that is, but it's something we're gonna, try.
Well,
There,
I would think that if you wanted to have the eDNA work as a tool, then it should eventually also work in state parks, right, or in any ecosystem, right? Otherwise, you're doing all for nothing. But
Right.
so, I mean, one thing that I've been thinking about a little bit is, what is the detection? What's the probability that you actually detect something when you look at ecosystems like a chrono sequence of invasion? So, if the invasion has just happened, would you be able to detect it? How many samples would you have to look at to detect it? And then as you progress in the invasion, if you have a really good year with hundreds of them per square meter, then is that leading to some kind of detection with the eDNA? You would think so, right? Um, so I think it's like a nice clone sequence would be cool. So if anybody has any sites where you know that they've just invaded, or where you have a really good invasion front, so that next year they're going to be maybe 5 meters along, then you can do before and after, right? So before and after the invasion happened, um, that'd be kind of cool. And I also think that um, the eDNA in a mesocosm, it'd be kind of cool to do like different loading rates of creatures, right? So if
Yeah,
you,
that's was the other thing. Yeah, we were going to test a couple different worm densities to kind
yeah,
of get at that. Yeah.
yeah, that'd be really cool.
Yeah, hopefully it'll work out. And then, yeah, once we have, a decent method, then exactly, it'd be great to get soil samples from different sites where it's known kind of when the timeline for invasion. Because down here we don't know anything. I know in Minnesota, just from talking to some of you and other people, there's sites where, they have a decent idea of kind of when invasion started and they're reliably there. So that'd be kind of a next level. Yeah, once we have a decent method, eDNA method, definitely do that.
We also were doing a lot of public outreach. That is kind of how I got started with all of this. I love educating the public, and I do a lot of work with the Land Trust of North Alabama. So that is one pathway to the public. I also have ties with the Master Gardeners, Master Naturalist, and Wild Ones of North Alabama, which is a nationwide group, but they focus on native plants. So I have really large groups of people who care about these things, and I'm hoping that in educating them and introducing them to iNaturalist, we can start seeing more jumping worm IDs on iNaturalist down here in the South.
So it's Angie Gupta again, and I'm just gonna jump in and say I've worked a lot with many of those groups, and I'm gonna drop in our link to Extension's eco-anxiety materials, because it turns out jumping worms evoked a ton of anxiety, eco-anxiety within our gardener community. So just heads up, it's going in the link.
Thank you so much.
Yeah.
So Katie, you're not at a land-grant university, so you don't have easy access to master gardeners and master naturalists, which are two groups that can help you. But
I
they
am one of each. I have
Oh,
done
okay.
the
I was going
courses.
to say they have a very clear hierarchy, and you can contact them and then get involved in the whole process. So to help you, surveys are really good. So you might want to try a survey about this, and you can publish it too as part of your thesis. But just try to figure out maybe anxiety issues or how many people think they have it or why they think they're a problem or whatever. So you might want to add that because we don't have a lot of information on. And again, we're trying to get this quantifiable effects of invasive jumping worms, so that we can use that in grants to get more money. Because right now we don't have a good way to a data-rich way to show they have economic and environmental impacts. So a survey that you did, all those surveys really do help a lot in quantifying people's perception and need. Lee, you've been really quiet. Can you tell us what you've been doing?
Well, I've been collaborating with Kiyoungsu and um, on the soil erosion project, with the grad student Nate London looking at erosion pins and I think cesium-137 and ground-based LIDAR to compare erosion rates with jumping worms and European worms, and we hope to establish some sites in Upper Michigan to compare almost earthworm free with European worm invasion. I mean, there is no earthworm free anymore now that we know Dendrodrilus rubidus is really Bimastos rubidus and that it's native from Vermont and Maine all the way across to northern Minnesota. But those worms are at a really low density, so it's almost earthworm free, and they're not the ones that eat the forest floor either. So we can't find a place where we have jumping worm invasion and European worm invasion and the almost earthworm free or kind of the pre-European condition all right next to each other, but we can do the two-way comparisons. Maybe Vermont has a place where all three are adjacent to each other.
So can I jump in? So Lee, I've heard you say that a couple of times about the new discovery of a native worm. Is that like, is there a peer review paper about that? Before we start taking down that there are no native earthworms in the state of Minnesota, I'd love to have that like documented why we changed our mind.
There are a couple of papers about it that have been published, including one that was recently published in the natural history section of Ecology, showing that they have an association with moose droppings.
Can somebody do you have those? Can you drop those in for us or?
Yeah, I don't think I can access them right at the moment, but I do. There was a preliminary paper, and then there was this one showing what it does in the natural environment. And again, it used to be Dendrobana rubidus, and then it was Dendrodrilus rubidus, and now it's Bimastos rubidus. So I call it the unhappy species. It shifts to a different genus every decade.
It's really interesting because, with the papers on global warming and the papers about this in Finland, where they found Dendrobana Bimastos rubidus, it really brings up this question: is there some kind of exchange between Finland and America, the US? I mean, the North America, which way did this thing travel, or are they actually two different organisms?
Yeah, I'm not sure that we know, but
well, we don't know. I know. I'm just throwing that question out there. I think it's an
yeah.
interesting question.
Somebody asked about glaciers, and obviously this native earthworm got back into glaciated areas. I mean, there's no reason that earthworms are excluded from areas that were glaciated. Michigan was almost all glaciated, and they have a whole bunch of native earthworm species in Michigan.
Yeah.
So, I mean, that relationship is not strict. There are a lot of places that aren't glaciated that don't have any native worms, and there are places that are glaciated that have the invasive worms, and it's really all over the place because different species travel at different speeds, and some of the native species have made it hundreds of miles into glaciated territories, and others haven't, just because they travel so much slower. You know, just like trees, aspen and birch trees are into areas as soon as the glacier leaves, and bald cypress is just still starting to respond to the fact that the glaciers left 17,000 years ago. It's just a very slow migrator, and the same is true of earthworms.
Yeah, I think Tim McKay showed that some of the one of the species of native earthworms south of the last glaciation in New York traveled north with the rivers draining into the lakes. So I mean, hydrophoria is like a big thing, and probably allows like for miles and miles of travel at a time.
Yep.
Which is cool.
Yep, and there was a paper just published about traveling in rivers up in, I think it was Manitoba, or it might be Saskatchewan as well. And we know that some of the native species have made it all the way up to the northern tip of lower Michigan, and that's several hundred miles. So that it's not a strict relationship between glaciation and the presence of native earthworms.
Yeah. Angela, you have your hand up.
Oh, I'm sorry. That's residual from my question for Lee. Sorry, it's down.
So Lee, I'm curious. In states where there are native earthworms and where there are jumping worms, is there less of an environmental impact on jumping worms? Is there any idea that if you have a community of earthworms, somehow there's something going on that mediates jumping worm damage?
I don't think so. I mean, the earthworm invasion fronts that I've seen in the Great Smoky Mountains, where they have more than a dozen native species, looked pretty much the same as the ones in northern Wisconsin and Minnesota to me. The native species are just functionally different. They don't completely eat the organic horizon and have all those impacts. They're not as dense in their populations. Invasive species are usually much higher density there, and the European worms are way higher density here than in Europe. According to my soil biologist friends from Europe, who can't believe how many lumbricus we have in the soil here. So the things that control their population in their native habitat and keep the population low so that it's in balance with the ecosystem in their native habitat just aren't here for either the European worms or the jumping worms.
Yeah, what Lee says for the European earthworms seem to be also true for jumping worms, because I travel to South Korea once a year, and then I travel in the countries. So all those forests I've been to in South Korea have Amiscus agrestis, but there are still thick horizons in well-protected forest. They don't have the forest floor that we see in the jumping infested forest in Minnesota Arboretum or state parks.
So is something keeping their, do you see the same large populations or are the populations smaller? So are there some keystone somethings that is managing their population so they're not as destructive?
Possibility, yeah. And I
Yeah, and
definitely I see less number of jumping arms in the forest in South Korea than I see here.
and those things could be nematodes that are parasites on earthworms. They could be fungal or bacterial pathogens. They could be things that exudates coming from the roots of plants. It could be a combination of a whole bunch of things that keep their population in balance with the ecosystem on other continents where they're native. And those are very complex and they're not known, but we can presume that in several hundred years, evolutionary processes will lead to controls on the invasive worms here, but it's not going to be in our lifetime probably. I mean, it would be nice if we were so smart that we could figure out that process and try to speed it up, but we're nowhere near. I mean, if there's anything I've learned in my career, it's how much we don't know. We're not anywhere near being able to figure out something like that, like how to get a new invasive species to be integrated into the ecosystem function and in balance with it.
And I think this actually raises an important questions for funding mechanism too. That, Vera, you earlier mentioned that invasive species may become one of the top priorities for a NIFA grant. But to really understand the underlying issues of the jumping worms, for example, we need to know why they do the things they don't really do in their homelands, and why do they why they become so numerous here, and those things need some international collaborations. And for jumping arms in collaborations with scientists in East Asia and European also, some collaborations in Germany and Northern and Southern Europe. So I hope that we can also talk to other people about this need and how the funding mechanisms can help those needs out.
Yeah, and I have some potential collaborations with some people in the Czech Republic to do some of those comparisons for the European earthworms. But that depends on funding from the Czech Science Foundation. So, yeah.
Anybody have anything to ask Lee? Thanks, Lee. Jim, do you have something you want to talk about?
I don't know, do I? Uh, um, obviously, the nursery and landscape folks are concerned about this and about spreading jumping worms. So we've been involved now for quite a few years. They're all looking for potential controls, and as you know, there are research projects going on looking at that, including the LCCMR grant that Vera has and others that are working on it. I don't know if you want to mention this now, Vera, but we've kind of run into a bit of a problem with a product that's shown up recently that's called jumping worm control, that is currently being marketed and people are finding it. But there are concerns about that particular product and what it really is. And according to MDA, it is a product that would be illegal to sell and to use in Minnesota as well as the entire United States, according to MDA. But that's true of tea seed meal as well. And we know that people are using tea seed meal all over the place, kind of like it's candy, for the control of jumping worms. So Vera and I were talking the other day, and I think an important fix would be able to get pea seed saponin onto the EPA allowable list. And it's frustrating to me why it's not there now, because it is used in food. It is used yeah, as coatings on fruits, strawberries, and things like that. So I don't know how much EPA has looked at that, but we're going to attempt to try and reach out to them again and find out where they're at with that. That's I think all I'll say about that, Vera. I can also say that I know that there are some new finds in some nurseries in the state. I know that they are aware of it. I know that some of them are using tea seed meal. Um, so there is an awareness out there, but the concern is still growing.
So there may be a magic bullet coming down the path. So Jim was telling me that Syngenta is trying to register a product with a different saponin from a soapbark tree in Peru. And they think I looked on their website and they say sometime this year they're going to have a regular registration for this product. And Garima has been doing work in our lab. At first, we didn't get the Kihia to work, and she's going to give you I know we're going over time here, so if you have to go, please go. But that may be the silver bullet. Is Syngenta is registering a product. They say it's going to come out this year, and they say it works against jumping worms. And so, in anticipation of this issue of why soapbark saponins don't work and THC male saponins do work, we started some research at higher levels. And so Green is going to talk about that in a second, but if you have to go, please go. So basically, we got, like Jim said, we have a little problem going on with a company who got our data, or I gave it to them. And then there were some changes in what's on the label, and they're trying to get people to use it. And it's just a mess right now. So we can't even really talk about it. It's just in terms of getting a product labeled, we're not there yet.
And we should say too that they did have an existing product, which was for controlling slugs, which works, but it's TC meal based. So that we still have the TC meal problem, and that we can't recommend it as a pesticide.
So this lab makes a liquid out of it, and it's 66% saponins compared to Planet Turf tea seed meal, which is 15% saponins. And so it's much stronger, and so you don't have to lug around the same bulk. So people would like the product, especially if you're at an arboretum and you want to kill some jumping worms that are messing with your hostas. So it has a lot of viabilities because of that liquid and the higher concentration. But unfortunately, it's not their fault, it's the EPA's fault for not having tea seed meal saponins on these 25B list. But maybe this Syngenta product will come out later this year, and that one they are looking for a regular registration, not a 25B. And so if they do get that registered, they should be a product out this year.
So I have a different kind of unrelated question for Jim. So Jim, I recall that at some point, like a poster was made, I think, for jumping worm outreach to your audience, and I'm just curious to know how that went and if it was well received and if if they've gotten it. Any of those questions?
Actually,
Thanks.
Laura put it in the chat.
Oh, great.
There's a link to the poster. We did distribute it to uh, garden centers and growers. To be honest, I haven't looked at Bachman's to see if they have it up or not. But I know they ordered posters for all their garden centers. So
Thanks.
So just to help understand about these three different saponin products out there, Graham and I found a paper the other day. Do you have that? I can share. You can just show the LC50s, and that will explain clearly what's going on with these different saponin products. So, Anna, can you share with Grima?
Yeah, I'm sharing that paper. Yes. This is the paper from Jiang and 2018. So they worked on different types of saponins obtained from three sources mainly. And if you see this table, so they have three types of saponins from Coelea saponins, that is soapbark tree, then tea saponins, and quinoa saponins. And if we see this is a lumbriculus, that's a nematode, and they compared their LC50s there, and the T saponin has 1.95, the clear saponin has 40 value, and then the quinoa saponin has 39 value. So this 85 value. So this is a good paper which they have three sources of saponins and they compared them, like the most effective ones are T saponins.
So that those are LC50, so the lowest LC50 is the most toxic. And
Yeah.
so that's why for us, when we tried initially we tried, there is a product called Monterey Nematode Control that has soap bark saponins. We tried it at about 10x the label rate, and now we've tried it at 100x, and we're getting around the LC50, and it is killing them. So I think that's probably Syngenta product is going to work. And as I said, it may be the silver bullet. Just you need much higher concentrations. Any other questions? So we've been over time and a lot of people stayed and that is wonderful. So we sent out this list of speakers and we sent out information on how we're going to organize this online workshop. And Joseph saw it and Brad saw it and now all of you have it. We want to get in some form. So please look at it and make suggestions. What we're going to do is we're going to interview people, have them give a PowerPoint presentation, we'll video it, and then we'll make a paper product out of it, we'll make a manuscript, and then we'll eventually put this online into an online course that any state somebody can come and take it, and we'll have a little quiz for each talk, and then we'll have a certificate you get so people can take the workshop and get credit with their state agency or their private agency. And so that's our plan. So Jumping Worm Symposium, Workshop, Conference, whatever we end up calling it. So we will contact people through the Healthy Soil Cooperative, and then we will ask them, what they want to talk about. And we have 4 or 5 categories that we want to have talks in. And then, like I said, we'll put it on the web in a format that anybody in any state can access it and take the course. So we're hoping to get this done and online by the fall. So we will send you a letter again with the symposium suggestions, and anybody can edit it and make suggestions. Does anybody have any questions on that?
Hey Vera, it's Laura. Yeah, I just asked in the chat, do you have a price point in mind for what this will cost for people to access?
We all can suggest that. The only reason we want to make some money is we want to publish all these talks in a journal. And journal articles now are around $2,000 a paper. We can probably get it down if lots of times these journals now want a whole issue devoted to a topic. And so we can probably get it down to 1,200 or so, but per paper. So we're hoping that the money we earn can go pay for the production of the written document. So how much for a person? That's up to us all to suggest. I'm hoping it's not over $50 for somebody to get a certificate. I don't want any money myself. We'll put this through the Healthy Soil Collaborative, and that's what I'm thinking, to make it as cheap as possible. But the revenue we do need to get to pay for the publication costs for the papers.
Yeah, that's super helpful. Thanks.
All right, well, thank you. If there's no other comments, suggestions, you know where we are. So just send us an email and Anna will be sending out through the Healthy Soil Collaborative more about this symposium. And this is a group effort, so we just started it. So we're interested in all your opinions and how you want to modify it, and we can talk more in written form about it. So please participate if you're interested.
Yep. If you guys have any comments, thoughts, questions, I'm a good resource to contact as well as Vera.
And I just wanted to throw out, since we're talking about talks, I just got another reminder this afternoon from the Upper Midwest Invasive Species Conference on their abstract submission. So just another plug for putting some talks into that. Thanks.
Awesome.
All right, guys. Well, you know where we are, and see you in 2 months then.